and talk about mutation as well, but if we don't, that's okay. We'll postpone it for on Monday. Today's Wednesday, is that right? On Monday, yes.
Put this on DocuShare.
It is on DocuShare. I just put it in last night. Chapter
I
7.
just checked.
Somebody asked me, somebody emailed me and said, okay, it's on DocuShare now. No? Okay, I'll do it after class. I thought I did it yesterday morning. And 17, I put 17 in there. But okay, I will put it on DocuShare, sure. But anyhow, so you know the basic principle of transcription. So does everybody know what I'm talking? What this chapter is about? You have to, anytime you want to study anything, guys. Maricela, anytime you want to study anything, try to understand the big picture, and then you can go ahead and study the details. Am I making some sense a little bit? What is the big picture of this chapter? That's your homework question. What is the big picture of this? What is this entire chapter going to teach us? I mean, including me. Anybody? Open floor. Transcription, translation. What do they mean? Tell me that you're talking about if this is the big picture, what you told me, and I don't mind to take just a few minutes off and talk about this. This is the big picture. And you told me transcription and translation. That's transcription, that's translation. Of course, there are other parts in this chapter too. So this is transcription. This is translation. What is the big picture? Anybody? Maricela. I'm picking on you today because you're daydreaming too much. I've been doing this for 30 years. I can look at your eyes and I know what's in your mind. And of course, when I meditate, you know, I focus here. When you focus here, then you can read people's mind. Not nice, but I can read people's mind. Evita, yes? Yes, sorry, non-read, I'm sorry, yes.
The gene expression?
Gene expression, what does it mean? Yes, but we didn't talk about gene expression. But what does it mean? Tell me, if somebody grab your collar in a mall and then shake you up, Stephen, and says, what is gene expression? What would you say? Charm? Anybody? Yes, Katrina.
It's the process for which DNA directs the synthesis of proteins.
Yeah, you're all direction of protein synthesis, direction of protein synthesis. Yes, all of that is good, but if you say that to somebody in the mall, the process of direction of protein synthesis, the problem. Steve, not to you, Katrina, to Stephen. Punch him in the face. What is it? Anybody?
Is it? This is RNA based on DNA. So you have your DNA, and then you have this different like, and then you top.
You guys are looking at your notes and giving me the answer. I don't want you to look at your notes. The answer that I'm looking for is not in your notes. The answer that I'm looking for, it is in these two pictures.
Oh, genotype and phenotype?
No. Somebody grab your collar in the mall and then
Mutation?
it will, no. Yes.
The difference between two different animals or like species, like how DNA has to change between.
We are almost getting there. Almost. Yes.
I would guess, well, like, I guess your body using your genes to like translate that into like physical appearance, I guess.
Is that phenotype?
The first two parts, your body get the gene and the kind of mom will beat you up. And then translate and beat you up more. And then you said what was the last one?
use that to, I guess, make your physical appearance.
And then he grabs you and hugs you and kisses you. Then he grabs you, hug you, and kiss you. Marcella, not you. Very good. That's it. That's all I'm looking for. What this chapter is all about. How in a cell you have DNA. That DNA determines you have black hair, you have brown hair, you have red hair, you have blue eyes. That's what this chapter is all about. Am I making some sense? That's how this chapter is determining. That's how this chapter is determining that your complexion. Is it white? Is it brown? Is it black? Am I making some sense? How tall you are? That's what this chapter is all about. All of the nuts and bolts. All of the nuts and bolts of determining your on top. All of this you said is correct. But the one that I'm looking for, your physical appearance. Physical appearance. That's what this chapter is all about. Then on the dinner table, you can impress your mom, dad, friend, whatever, and talk to them about these things. And then say, oh wow, translation, and you teach them about transcription. You know, the best way to learn the subject is by teaching it. That's what they say. So it's teach your little 2 years old, 3 years old at home, 5 years old. Come here, sit down, and I want to tell you all about transcription. Yes?
Isn't that the same thing as transcription?
Yeah, physics. But if you go to the mall and you're in a movie theater or in a grocery store, somebody grab your collar and ask the question, you say phenotype. Not you, but they pump. They pump Stephen. Phenotype, what does that mean? You and I know, but somebody in the mall does not. Am I making some sense? So the big picture in lame language of this chapter is what is the physical appearance, which is phenotype. And Stephen finally came to our rescue. Am I straight? I know I'm straight, but am I making some sense? Am I making some sense? Huh? Am I making sense? Everybody, let's go. So what happens? We talked about this last time. Transcription, of course, messenger RNA, translation, ribosome. All of those protein molecules that determine your complexion, the color of your hair, blonde, black, brown, red, color of your eye, all of those are protein molecules that determine that. Am I making some sense? And who tells that protein molecule for blue eye to be synthesized? Your genes. Is that right? Your genes. Transcription. Transcription is when RNA is synthesized based on DNA. Quiz question today. Okay, so you had it last time. I don't know. But anyhow, mRNA is the messenger. We'll take it to cytoplasm in case of eukaryotic cells and tell, okay, now make the protein molecules for blue eyes. I always refer to blue eyes, blonde hair, dark hair, so you relate what I'm talking about, what this chapter is all about. Okay, am I making some sense a little bit? Good morning. And then the location, ribosome, that's where all of these amino acids or protein molecules are made up of. Chapter number 1. First exam, not chapter number 1, first exam material. Proteins are made up of what? Huh? Huh? Nobody knows? Let's go back to chapter 1. Examine. Huh? What is it? Protein. Amino acids. Please say it loud. Don't worry. And the answer is no. I say no. So what? Will they take some of your gold away from you? You all have golds. Money, I guess. I have any. I do not have any. You will not be charged. Okay. So how did we get there? I guess my hand. So anyhow, translation ribosome. In prokaryotic, translation of mRNA begins before transcription. Right here is the central dogma of biology. We talked about this. That's transcription. This is DNA, and RNA is built. DNA is double-stranded, right? RNA is single-stranded, and then RNA is synthesized based on DNA. mRNA in this case comes out in cytoplasm and goes through ribosome and make up the amino acids, the blue color, the blue eyes, right? Blue eyes. This protein molecule right here is the blue eye. I want to make it some sense. The end product, you know the end product. So the rest of today, the next hour or so, we spend time all the way from here to here. The steps, what is involved, that's what the next hour or so, you and I, you woke up this morning, you drank coffee or not, tea, whatever, you got up, took a shower, whatever you've done to come here to listen to this. Is that right? Yes? Or if you choose not to, you could have go to work, do something else in life. But you decided in life, you decided to come here and listen from here to here. What happens? Am I making some sense, everybody? Might as well. Pay attention. Your body is here, right? Maya, your body is here. Bring your mind here too. Please. Are you guys ready? Shall we move on? Okay, and then of course in bacteria there is no nucleus, you know that. So the transcription and translation can happen simultaneously. It can happen. While transcription is occurring, translation is occurring here too. I hope I'm making some sense. So you're familiar with those two terminologies. If you're not familiar with any terminology, any word I threw at you, please stop me, send me, what does that mean? And if I don't know, we Google it. We go to dictionary.com. And we'll come up with the answer, or ask classmates, what does it mean? Okay, but transcription, translation, I'm giving you those right now. Let's go. A primary transcript is the initial RNA transcript for any gene prior to a processing. It means that that's the primary transcript right here, and then you have make RNA, and RNA based on that is protein. That's the central dogma in biology. The central dogma is constant at all cells. Okay, how are instructions for assembling amino acids in proteins encoded in DNA? There are 20 amino acids. You already know that. Is that right? You and I are made up of 20 different amino acids only. And totally, if they take, Amir, it's only 20 amino acids. Yes, it's only 20 amino acids. They take your kidney and put it in mine. Will I accept it? No. It's only 20 amino acids, Amir. No. The book that I write is totally different than the book she writes. Am I making some sense, everybody? How many letters in English language? I should ask this. Huh? No, I should ask my daughter. How many letters? You all know this. Oh, 20? 20 what? Kimia, how many? 26? Okay, I don't know, 26, 24, something in that neighborhood. How many letters in English language? 26. How many words in English language? Don't say indefinitely, unknown. No. We talk to a linguistic. How many words? Brian, how many words do you think there is in English language?
They're still making them.
They're still making them. I love that. Yes, you're right. They're still making them. But I talked to a linguistic faculty at Delta College. He used to teach language. About 500,000. And English language has 500,000. You open up a dictionary, it has about that many, up and down. As he said, they're still adding it. And that's true. Now, are you ready? 26 letters. 500,000 words. Are you ready for next question? How many books in English language? Who knows? Indefinitely. Could be millions, billions, trillions, and still more coming up every day. Which one do you think is more? Words into English language or books in English language? Books, definitely. Every day, there are hundreds of them being published all over the world. I'm talking English language, not only in the United States, all England, Australia, Scotland. Am I making some sense? So the book that I write, is different than the book she writes. Am I yes or no? 20 amino acids, 26 letters in English language, 26 letters. How many books? This is a book. Imagine me as a book and imagine her as a book. That's why I would not accept, let's see, you might think, oh, it's as a woman, I would not accept his kidney, even though we are same sex. I want to make it some sense. I would not accept his kidney because of those 20 different amino acids. I want to make, if my were identical twins, if we were identical twins, would I accept his kidney? Yeah, of course. There are 2 copies of your textbook. Imagine you have 2 textbooks exactly duplicate of your textbook. I take out 1 page and put it in the other textbook. Same, nothing will happen. But if I take a Shakespeare page and put it in your textbook, everybody would know. Am I making some sense, everybody? So that's the idea. We have 20 amino acids, but at the end, somebody has red hair, and somebody has blonde hair, somebody has black hair. I hope I'm making some sense. Yes, let's go.
And what about like organ transplants?
That's what I said. That's what you're talking about. Organ transplants, I will not accept it.
Yeah, but like, don't some people, cause like that's like you can get that like.
Yeah, it does. Of course, with medication, and they look how closely. If I grab 2 biology textbooks, 2 different authors, okay? I grab 1 page from protein synthesis chapter and put it in the other textbook, it might fit. It stands out, hey, I'm different, but it might fit a little bit. But if I take Shakespeare book, if I'm Shakespeare and his biology textbook, who would not? But if I'm a biology textbook, he's a biology textbook, we match. We have to trim the pages a little bit. Maybe my page is bigger. Or with medication, they allow, transplants. People who have transplants, most of them have to take some kind of medication, okay, to repress the immune system. I hope we spend half an hour, oh my God, half an hour just to give you big picture of what this chapter is all about. Am I making some sense a little bit? And who determines this? DNA, of course. So DNA determines it. So 20 amino acids. And then how many nucleotides correspond to an amino acids? There are 4 nitrogenous bases. You remember adenine, thymine, guanine, cytosine? Those 4 determines what you are. How does computers work? All of the computers on planet Earth, anybody on computer plan, designing computers, chips, programs, anything is based on 0 and 1. And how many new programs is coming out every day? How many video games is coming out every day? Zillions. And it's all based on 0 and 1. Right? Am I making some sense? So you and I are different based on 4 letters. Those 4 letters determines my hair is black, my eye is brown, her hair is red, her eyes is blue. Okay, let's go. Right here, pretty much DNA double-stranded, and then the DNA determines what the RNA is supposed to be, mRNA, and mRNA determines, yesterday you could not see this. Can you see this, everybody? These are not in the way, I hope. And then this is the amino acid, tryptophan, phenylalanine, glycine, and serine. These are the amino acid. Translation, it means based on the RNA, protein, amino acids are going to be synthesized, and based on DNA, RNA is going to be synthesized. A lab you're going to do today. That's what you're going to do today. In the lab. Right? Nice fellow. And then, of course, this is the table. I hope in the lab, I'm not going to go over it, but in the lab, you're going to need that. If we cannot put it up, then grab a textbook. The whole entire table is in the lab. Last time this was not, Marisa, you were not here last time, but right here, well, I was up to here, but the whole thing is up there, okay? A couple of things before I move on, because we need it. AUG is the signal to start protein synthesis. And then these 3, UAA, UAG, and UGA, these 3 are to stop the protein synthesis. Okay, I hope I'm making some sense. Oh, there are 64 codons. This is called a codon. This is called these three nitrogenous bases called codon, 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 codon. Yeah, if you look at this table, there are 64 of them, and you should know how to read them. The first letter U, the second letter U, the third letter U. So it's U, U, U. It means phenylalanine. Amino acid phenylalanine. Okay, and then this one C. First letter is C. Second letter is A. Third letter is A. CAA is glycine. No, that's not glycine. That's glutamine. Sorry, glutamic acid, glutamine right here. That's glycine. Anyhow, so there is one start signal which is methionine. So all protein molecules starts with methionine, amino acids methionine, and then all protein molecules at the end must have a stop signal. Say stop. So 3 stops, 1 start slash methionine, 64 codons all together. Let's go. The genetic code is nearly universal, shared by simplest bacteria. Even bacteria has 4 nitrogenous bases. Bacteria have different protein molecules than you and I. They have different amino acids than you and I. So the 20 amino acids does not fit the bacteria scheme. But the 4 nitrogenous bases, adenine, thymine, guanine, cytosine, fits bacteria too, fits fungi. Anything, any living organisms on planet Earth has those 4. Okay, genes can be transcribed and translated after being transplanted from one species to the other, which we did talk about this. Okay, molecular components of transcription, RNA polymerase. Let's start. So the first one that we are going to talk about is transcription, based on how DNA, this is DNA, double-stranded, right? Based on DNA, and RNA is going to be synthesized. Based on DNA, this is called what? Transcription. Okay, so what is involved in transcription? The enzyme RNA polymerase, ACE at the end of the word. What are the 3 functions of RNA polymerase? Okay, number 1, makes RNA, of course, it makes RNA right here. That is made by RNA polymerase and does not need a primer. You remember when DNA was going to duplicate? You remember that? Last chapter, this was DNA unwinds, trying, and then a piece of RNA had to be initially started. It was called primer, and a primase was the enzyme responsible for that. We don't need it here, not in transcription. We need it for DNA replication, but not for transcription. Okay, so makes RNA, does not need it. Opens up the DNA. The DNA, of course, has to be opened up. If this is DNA, has to open up, that RNA be synthesized next to it, to this arm, to the template. You remember that? We have template, and then in your handout today for the lab, we call it sense strand or template. This is a nonsense strand. DNA is double-stranded, so this is a sense strand or template, then RNA is going to be synthesized next to this one. I hope I'm making some sense. Okay. And then pair up uracil with adenine. Of course, in DNA, right here, in DNA you have adenine, you do not have thymine. In RNA you have instead uracil. You remember that? This is thymine, this is adenine, so this is uracil, uracil. This is guanine, this is cytosine. This is cytosine, this is guanine. It has to follow the law of base pairing. It will follow the law of base pairing. The DNA sequence where RNA polymerase attaches is called promoter. So right in this area is called a promoter. I'll show you some pictures. In bacteria, the sequence signaling the end of the transcription is called terminator, which comes up later on. Here is a promoter. That's a DNA strand. Here is a start point. right here. So my promoter would be right in this area, right before it starts transcription. So the start point is right here, and this one is promoter. So the RNA polymerase right here, it will attach to that promoter. And of course, the transcription occurs right here. What else? Initiation, you have the RNA transcript unwound, the RNA polymerase. I did an RNA polymerase. First function, unwound DNA, and then of course starts the RNA. And this is a template, and this is the nonsense strand. This is the sense strand. Elongation. So there are 3 stages in transcription, and you will see the same 3 stages are in translation: initiation, elongation, termination. Those 3 stages of transcription are initiation, elongation, and termination. So what happens during initiation? Of course, DNA has to unwind. RNA polymerase comes in place and attaches the promoter. Promoter is a DNA sequence. So RNA polymerase recognizes that, and then, of course, RNA polymerase unwind DNA and starts synthesizing the RNA based on the template strand. And then what happens during elongation? Pieces and bits of the RNA attached to each other, little by little, little by little. There is one word that later on, if you're going to go to research or you're going to study biology a little bit more, this word will come up: downstream and upstream. They call it downstream and upstream. I'll put a star next to it. That's an important term. It means the sequence of events that is happening. It means transcription is occurring that way. Okay, of course, at 3 primes, it has to happen at 3' end. The RNA is being added at 3' end, but that is downstream, right here. Upstream would be here. Okay, so that's a terminology you might encounter. And at the end, when it's all over, termination, look what happens. The DNA comes back together, the same original DNA, and then RNA polymerase separates, and then right here you have your mRNA. That's it. That's termination. It's over. Transcription is over. Let's go a little bit more detail. Again, I'm talking about transcription. A little bit more detail. Animation. Okay, let's see what animation has to say.
The first step in protein synthesis is transcription of RNA from DNA. The portion of the DNA that is transcribed into an RNA molecule is called a transcription unit. An enzyme called RNA polymerase carries out transcription. With the help of protein transcription factors, it attaches to the beginning of a region of the DNA called the promoter, pries the DNA strands apart, and untwists a short portion of the double helix. RNA polymerase moves along the DNA, pairing up RNA nucleotides with their DNA complements, adding nucleotides to the end of the growing RNA molecule.
Come on, I never learned this, huh? Okay.
The first step in protein synthesis is transcription of RNA from DNA. The portion of the DNA that is transcribed into an RNA molecule is called a transcription unit, called RNA polymerase, carries out transcription. With the help of protein attaches to the beginning of a region of the DNA called the promoter, pries the DNA strands apart, and untwists a short portion of the double helix.
If I want to pause it right here, right?
RNA polymerase moves along the DNA, pairing up RNA nucleotides with their DNA complements, adding nucleotides to the...
Do you know how to pause this?
The first step in protein synthesis is the double helix.
Okay.
Nucleotides with their
Okay.
DNA complements.
What he's doing,
Adding nucleotides
look, this
to the end of the
comes
growing
back
RNA
together,
molecule.
and this will go
Here's a
forward. So
close-up
you don't
view
need the
of
enzyme
elongation.
topoisomerase.
Only one DNA
You don't need
strand,
the, you all know
template strand,
topoisomerase?
serves
We
as
talked
a
about it last
template
time.
for
You don't
RNA
need it.
synthesis. RNA polymerase moves along the DNA strand in the 3' to 5' direction,
3'
adding
to 5'.
nucleotides to the 3' end of the RNA chain. Note that U in RNA pairs with A in DNA.
Okay.
Once transcription is complete, RNA polymerase releases the completed RNA and detaches from the DNA.
That's elongation. And now termination.
Then there's an mRNA.
That's mRNA. Other RNAs can be synthesized this way as well, but that's pretty much mRNA. We are focusing on mRNA. The other two, tRNA and ribosomal rRNA, there are three different types of RNA. mRNA, which is messenger RNA, transfer RNA, tRNA, and rRNA. This is the mRNA.
What? You just said four.
Three. mRNA, tRNA, and rRNA. And I will go over all of them hopefully today. Okay. So right here, quickly, and these are transcription factors, protein molecules that is recognizing and getting ready, setting up the, attaching to this DNA and getting it ready to go. Now, in the promoter, some promoter, you all know what promoter is. This is a start point. Everybody following what I'm saying? This is the start point, and this is the promoter. This is the downstream. This is upstream, this is downstream. See, I sometimes make mistakes. But what happens, what is a promoter in most cases, they call it a ta-ta box. First time I heard that, I laughed. I thought it was funny. Ta-ta, that's what you tell a little kid. Ta-ta, you know, don't you say ta-ta when they're walking around? No, everybody's different. Ta-ta box, I said, you're funny. They said, no, Amir, it's called ta-ta box. So I want to see it in print, and right here. So ta-ta box, it has, what does it mean? It means here, there's a lot of adenine and thymine, adenine and thymine, adenine and thymine, thymine, adenine, thymine, adenine, thymine, adenine, adenine, thymine. They call it ta-ta box. So there's a lot of TNA, and what happens, these transcription factors recognize that. When they recognize that, then RNA polymerase comes, and of course you know the functions of RNA polymerase, unwind the DNA and starts adding the 3' to 5' RNA molecules. Okay, I hope I'm making some sense. So eukaryotic promoter, that's a promoter. This right here, ta-ta box is a promoter. If you would, ta-ta box is a promoter. So multiple choice question, what is it? Which one of the following could be a promoter? GC, GC, GA, GC, GC. Or TA, TA, TA. TA, ta-ta box is a promoter. But anyhow, several transcription factors come in. These are protein molecules. Transcription factors are protein molecules. And then, of course, the RNA polymerase comes in and starts synthesizing. Elongation of the strand. So that was all initiation. Elongation, the next step, you remember initiation, elongation, termination of the transcription. We're talking about transcription. Right?
Initiation,
Elongation,
elongation.
and termination. Okay, so as RNA polymerase moves along the DNA, it untwists the double helix 10 to 20 base pairs only. So you don't need the enzyme topoisomerase. You remember in DNA replication, we talked about topoisomerase. And then transcription progress at a rate of 40 nucleotides per second in eukaryotic cells. A gene can be transcribed simultaneously by several RNA polymerase. That I'll show you a picture of that later on. So what you have a RNA RNA can be synthesized by RNA polymerase here, RNA polymerase here, RNA polymerase here, several RNA polymerase. That's what he's talking about. Okay, nucleotides are added to the 3' end. I already talked about all of this that growing to the molecule. Right here, this is your RNA polymerase, and here is the 3' end, and of course, you know, T, A, U. Remember that today on the lab? Please do not make mistakes. The sequence of amino acids today in the lab, you're going to turn to me. All of you should be same, because you're all getting same handout, right? So if somebody does not have the sequence, the right sequence of amino acids, I return it to you, say, fix it. So you have direction of transcription. It's going to have, that's downstream, is that right? And this is upstream. So we talked about template, and so on and so on. Okay, termination. So it was elongation, initiation, elongation, and finally termination of transcription. The mechanism of termination are different in bacteria and eukaryotic cells. Yes, of course. In bacteria, the polymerase stops transcription at the end of the terminator, and the mRNA can be translated without further modifications. In eukaryotic, however, RNA polymerase II transcribes the polyadenylation signals. And poly, it means what, guys? Poly means what? Many. Adenyl, it means adenine, 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 adenine. At the end of the mRNA in eukaryotic cells, you have several A. And that's what polyadenylation means. As Brian said, in English language, we add new words. This word didn't exist ever, but we are adding it. So, anyhow, the RNA transcript is released 10 to 30 from nucleotide past the polyadenylation sequence, and after this, still 10 to 35 more nucleotide is going to be added.
What's that called?
Polyadenylation.
That's just a word for the RNA polymers? Or how?
No. On the RNA, you remember on the RNA? On the RNA, right here, at the end of the RNA, right here, you have several A. That's what's called polyadenylation. At the end, this is termination. You're talking about termination.
That happens at the end of every sequence?
What?
That happens at the end of every RNA sequence?
Every RNA.
mRNA sequence?
mRNA.
So when mRNA is created, like the AAA is at the end of it?
That's the end of it. You know it's over, almost over. Yes. After this, there is no more amino acids going to be put together. I'll show you a picture in a minute. Yes?
What's the difference between RNA polymerase 1 and RNA polymerase 2? Is it the same?
There are two different enzymes. RNA polymerase II does function the same thing as it's usually toward RNA polymerase II. It's doing the job at the end of the mRNA. Okay, at the end, the transcription. Yes, that's right. At the end, termination. Okay. Alterations of the mRNA ends. Each end of the pre-mRNA molecule is modified in a particular way. The 5' end receives a modified nucleotide 5' cap, and I will talk about this, and you see it. And the 3' end gets a poly A tail. Okay, so the 5' you get a cap. So this is the 3' end. If I would, that would be a 3' end. That would be a 5' end, and right here you get a cap. And you will see it. That's what I mean. Okay. So these modifications say several functions. So why that would be a nice essay question. Why you add a poly A and a cap to the mRNA? Why you do that? The answer is they seem to facilitate the export of the mRNA to the cytoplasm. Again, you remember the mRNA is going from nucleus to cytoplasm. It has to go with that. It protects mRNA from hydrolytic enzymes. There are some enzymes can come and snip off, break down mRNA. So it protects it to some extent. These two tails will protect them. Or the cap, I'm sorry, one end, sorry, the cap is at this end. This end is cap. This end is cap. This end is AAA. So when the mRNA is going from nucleus to cytoplasm, right? So these two are protecting what is in between. I hope I'm making some sense. So the cap and the poly A, if there is an enzyme wants to attack the mRNA, this is mRNA, then this one protects it and the cap will protect it as well. And I'll let you know in a minute what is a cap made up of. Okay, hang on. They help ribosomes attach to the 5' end, and that's what's going to happen. Right here is the cap. You have a modified guanine. You all know what guanine is. It's a nitrogenous base with polynucleotide attached to it. Okay, so you have 3-phosphate, and that's a cap. Okay, guanine with 3-phosphate attached to it. What is UTR? You have untranslated regions. It's called UTR, 5' UTR. You have here UTR, you have here, and then of course you have your poly A tail. Okay, so that's pretty much is the structure of an mRNA. This is an mRNA. This is an mRNA. You have a cap, and you have a poly A tail right here. And what is the why they exist? Because to protect what is in between.
What's the UTR step? What's that?
The what?
UTR?
UTR, untranslated regions.
Oh.
It means these regions will not turn into amino, they will not direct amino acid synthesis.
Oh, so those don't have like any.
And I will talk about that.
GC, T good, just says nothing. But then it starts at the start. There's like GC, all the sequence.
Right. Right. Yes. Okay. So if you look at this, guys, this is the region of includes proteins encoding, where the protein synthesis is going to be right in this area. Do you all follow what I'm talking about? And then at the beginning and at the end, you have untranslated region. It means these regions are not going to direct any protein synthesis. Am I making some sense? Right here and right here. So Amir, this coloring, Brian is asking me, this coloration is same as that color, this is same as that color, this is same as that color. It means they will not be translated either. However, we have a different name for them, for these regions. For these regions right here, from here to here, you all follow what I'm talking about? From here to here, from here to here, we have a name for it, and that name is called, they're referred to as intron. Okay, so intron are different than untranslated region. Untranslated region is a region that is at the end or at the beginning of mRNA. But introns, which they do not be they are not going to be translated, they are within the protein coding regions. Let's look at it. I give you a prelude of what is coming up. Introns, non-coding regions. Non-coding regions. But this is untranslated regions. These are non-coding, which we refer to them as introns. And exons, translated regions. Let's go back. This is exon, right here. This is exon, right here. You see the different of colors? Do you guys see the different colors? So exons are the area that are going to be translated. Okay. Then I have to toss at you the term RNA splicing removes introns and joins exons. So that's the term that is used in some cases. RNA splicing is carried out by spliceosomes. That's a protein molecule. Spliceosomes are proteins and several small nuclear ribonucleoproteins, SNRNPS, that recognize the splice sites. Ribozymes, at the beginning of semester, I said all enzymes are protein molecule. I lied to you. I had to lie, right? But now I'm giving you one enzyme. There is only one enzyme that is not made up of protein. It's not protein molecule. It's RNA molecule. So ribozymes are catalytic RNA molecules that function as enzymes and can splice RNA. Ribozymes, it is RNA molecule. It's not DNA. It's not protein molecule. It's an enzyme which puts these exons together, attaches the exons. If I remove, are you following me? If I remove this, who is going to attach this to this? Ribozymes. Who is going to attach the exon to the exon? Ribozymes. Okay? What cuts off this and cuts off this is called spliceosomes. Spliceosomes will cut off this and that. But what puts them together is called ribozymes. I hope I'm making some sense. Okay? Here they are, cap region. Here is 1 to 3. This is the coding region right here. Intron, exon, right here. And the exon's coding segments. This is the coding segments with a cap and poly A tail. This will go from nucleus to cytoplasm to be translated. Right here is a spliceozymes. Spliceosomes is a protein molecule, and then inside of it you have small RNA molecules are being spliced, and at the end you have exon 1 and exon 2 combined together.
Can you go back a slide or two?
Sure.
For the ribozymes.
Ribozymes are catalytic RNA molecules that function as enzymes.
So what are they again?
They are enzymes. Ribozymes are enzymes, but what is their function? Their function is to put the exons together.
They put the exons
Together. That's it. That's what the function is. And here, this diagram will tell you pretty much what is happening. Here are the introns that they will not be translated, but the exons will be translated. Okay, so mRNA, exon 1 and exon 2,
So
and here are
polymerase will take out the introns, like they just cut them out.
there,
No.
spliceosomes.
Oh, the
Spliceosomes
spliceosomes will cut
will-
those out.
We'll cut them out, splice them out. Very good. Splice them out. Very good. Okay, the functional and evolutionary importance of introns. So one person might ask Amir, why the cell bothers to go through making introns and exons? Why just does not make exons and get it over with? Am I making some sense? Why do you make introns that they have to be spliced, cut off, and then trash? Why not? Here's the answer. Why? Some introns contain sequences that may regulate gene expression. Some genes can encode more than one kind of polypeptide, depending on which segments are treated as exons during splicing. This is called alternative RNA splicing. Consequently, the number of different proteins and organisms can produce in much greater than the number of. What am I saying here, guys? Can I erase all of this? So you know this chapter is all about physical appearance, which you were born with. Okay, so what am I saying? Again, this could be a nice essay question. Why you have these exons and introns? For example, let's draw our